All right, hello everyone. My name is Maddie Stover. I'm also a graduate student with the Luthi-Shelton group at Illinois. And today I'm going to be talking about modeling cell growth of SYN3A. So as my colleagues have previously touched upon or described, our group does a whole cell model of the minimal cell on a software called Lattice Microbes, which simulates the cell stochastically by sampling trajectories from the reaction diffusion master equation and chemical master equation. We discretize physical space on an 8 nanometer cubic lattice for SYN3A, which distinguishes different cell regions as different cell types, as I've shown in this figure over to the right. So here we have the extracellular space in this light blue, the membrane in pink here, the cytoplasm in purple, and the ribosome in yellow. And these white particles are allowed to diffuse between subvolumes and react within subvolumes. So we have this whole cell model, and we want to incorporate an entire cell cycle into the model. So to do this, we need the location of the ribosomes throughout the cell growth, which is given to us by cryo-electrotomograms from Elizabeth Vila. And we also need a growth rate, which is given by our lab's metabolic model for synthesis of lipids and membrane proteins and the incorporation of these membrane proteins via the SEC-A, SEC-Y system, which was discussed by my colleague David earlier this morning. So all that is missing to incorporate this whole cell cycle into our cell model is an actual algorithm for cell growth. So to accomplish this in Lattice Microbes, we'll have to update the following three things as the cell grows: ribosomes, membrane size, and the membrane-bound and transmembrane proteins. So before I talk about our model for growth of SYN3A, I want to briefly touch upon some previous work in our group on cell growth. So in 2016, as a part of his work on ribosome biogenesis, Tyler Ernest modeled cell division of E. coli in lattice microbes, which is shown in this figure down here. So gray is extracellular, green is the membrane of E. coli, orange is the cytoplasm, and in his model, Tyler begins with E. coli as some initial length. Here it's 2.4 micrometers, and the cell volume grows constantly throughout the cell cycle at an exponential rate, whereupon it divides into two daughter cells of the original length of the length of this original E. coli, so 2.4 micrometers. While both of these models for cell division are simulated in lattice microbes, there are a few important differences. Because of its rod shape, E. coli only grows in one dimension in Tyler's model, whereas SYN3A grows out spherically in all three dimensions in our model. Tyler's model is rule-based for E. coli, whereas our growth for SYN3A is based on the full kinetic model of the minimal cell. So in other words, in this model of E. coli growth, the programmer decides the rate at which the cell is going to grow, where our growth of SYN3A is dictated by metabolic conditions such as lipid synthesis and the corresponding growth in cell surface area. So again, just to quickly review, to grow SYN3A, we have to update ribosomes, membrane size, and membrane-bound proteins. So to update ribosomes as the cell grows, we begin with the tomogram of the large cell. And in this cartoon depiction down here, we have a membrane shown in this red ring, and then the ribosomes are these black dots. And in steps of 8 nanometers, we reduce the radius of the cell membrane to create 6 layers until you have reached the radius of the small cell. And then when we go into the simulation, we can then update the ribosomes by adding these layers of concentric spherical shells to work up from the small cell to the large cell. So again, just to show this with the tomograms, we have the tomogram of the large cell on the left and the small cell on the right. My colleague Benjamin discussed how we use template matching to get the distribution of these ribosomes, 820 for the large cell, 503 for the small cell. From this computational model, we can show the distribution of ribosomes throughout the cell. And specifically, these are the layers that I add during growth. So each data point on this plot is a layer of ribosomes that we add during cell growth. Going again from the small cell to this large cell, about 250 nanometers in radius. So as the cell grows, we have to update these cell region types that I discussed earlier. So the membrane will have to move out spherically by 1 lattice site. And if you can imagine a membrane protein like these particles in blue, as the membrane moves out, these particles will be now located in the wrong cell region, for example, the cytoplasm here. So it's necessary to move particles outward radially as the cell grows. So again, just a cartoon depiction of this. At time t equals 0, we have a particle in the cell membrane. We grow it out spherically by 1 layer. The particle is now left behind in the cytoplasm, so we have to translate the particles out along with the cell as it grows. So what is the computational method for implementing these? So running this RDME CME that's been discussed, we pause the simulation, check for certain cellular conditions. So for example, the surface area increase, the metabolic conditions such as lipid synthesis, and if these conditions are met, we will actually implement the growth algorithm. So update the cell regions and move membrane particles. And then this information gets fed back into the RDME CME, updates it, and then we continue to run the RDME CME. So some quick summary, we've been able to include a model for cell growth into the existing whole cell model and lattice microbes. And I'll just show a brief video of this. So the cell membrane is here in green, the ribosomes in yellow. These red particles are degradasomes, and then the orange is DNA. And you can see here that the DNA is not yet growing, but this is where we're going to incorporate Ben's work. So yes, and then moving forward, we want to speed up this algorithm and then begin constructing a model of FTSC formation and subsequent cell division of syn3A. So here we have just an example of a rod-shaped bacteria, subtilis, FTSC forming and facilitating that cell division. This work on FTFC is motivated by super resolution imaging from the Ha and Xiao labs, who will be giving talks next Monday. I want to acknowledge my advisor, my group members, and the JCVI for hosting. Thank you for listening and happy to take any questions.
Maddie, how long before you think you'll have this system all set up?
You mean in terms of the division?
Yeah.
I am not entirely sure. Hopefully we will get some kinetic parameters from the super resolution imaging. And we have a method for some filament modeling that we're looking at. So we're kind of exploring those options now.
So, you are aware of James Pelletier's paper where we identified 5 unknown genes that were involved in cell division. So if we can, based on Jörg Stolke's group and Yuri Rapsilber's group's efforts at looking at the minimal cell interactome, we may be able to get a clearer idea of possibly what these 5 unknown proteins of unknown function might be doing in cell division, and that might facilitate your modeling some.
Yeah, that would be great. Yeah, definitely.
Hint, hint, guys in Germany.